We're at the end of chapter 4. We've covered vomiting and diarrhea and how to make those happen and how to stop those from happening. So we're on to our last couple of learning objectives. We're going to talk about treating ulcers, mostly gastric ulcers, and we're going to talk about rumen motility and how we may need to help a ruminant to have their rumen work better. It's kind of the short way to put it. Um, so in the ulcer category, we're going to be talking about antacids and anti-ulcer drugs, and then a few other things. So we need to talk about how things happen in the GI tract before we can talk about how we're going to modify them with drugs. So ulcers, we've talked about a couple of ways that we can induce ulcers in the previous chapter in anti-inflammatories. So both steroids and NSAIDs act on to modify the formation of prostaglandins that are protective to the stomach. And so giving too much of those, or what's really bad is to give them both at the same time, they really clamp down on that production, and we don't get good mucus formation or release of bicarbonate, which is kind of right in that mucus, and blood flow to the GI tract. So those are definitely a potential cause of ulcers. Anything that would cause hyperacidity, bile reflux into the stomach can be very irritating. Any sorts of toxins that accumulate in that area, and just stress. Stress can induce ulcer formation. In ruminants, because the rumen is mostly alkaline, but there are conditions we tend to want to feed ruminants grain, especially if we're using them for milk production, in a feedlot where we're concentrating on the last few months of meat production, muscle buildup. A lot of those kinds of producers will tend to pour a lot of grain to these animals, and they were not meant, the rumen is not meant to handle that much grain. It causes an acidic condition. It changes the rumen microflora and can cause gastric ulceration. So hopefully these names are going to be familiar to you, but we're going to talk about the oxinic or parietal cells. And these are in the stomach lining, and they are stimulated to produce hydrochloric acid. There's 3 different receptors that we'll talk about that are on the cells. The maximum acid production occurs when all of these are stimulating the parietal cells. So there's histamine receptors. These are a bit different than the histamine receptors that we think about in allergic reactions causing redness and swelling in an area, edema in an area. And there's gastrin receptors, and we'll talk about gastrin in a minute, and then acetylcholine. So do you remember which autonomic nervous system acetylcholine is a neurotransmitter for? If you remembered the parasympathetic nervous system, then you're on track here. That's the logical one, right? When we're talking about digestion of food. So acetylcholine, there are acetylcholine receptors on these cells as well. So this is what the cell looks like. It has these 3 different types of receptors on it, and you can see that, if they're all activated, then we have the maximum amount of hydrogen being pumped out. Hydrogen is pumped out. It combines with chloride in the stomach, which is produced in another area, and that produces hydrochloric acid. Now, the stomach is very acidic, has a lot of these hydrogen ions in it. This is an active transport pump, so it involves energy because it needs to push against that concentration gradient in the stomach to pump out more stomach acid. So how do we get the things that act on the receptors? I'm kind of moving ahead to a slide. I kind of wish I had these in a little different order, but we'll talk about gastrin release first. So gastrin is released by G cells. It's produced by G cells, and it is in response to proteins in the stomach and stretching of the stomach due to the presence of food or a liquid. And it's all those things are signaling, okay, we need to start the process of digestion. So gastrin is released, and they act on enterochromaffin-like cells to release histamine. So the food goes into the stomach, presence of protein, and the distension of the stomach induce the G cells to release gastrin. Gastrin affects the enterochromaffin-like cells that then release histamine. And acetylcholine is the parasympathetic nervous system neurotransmitter. And when an animal starts to think about eating, anticipation of eating, time of day, or seeing something, smelling something, then acetylcholine is released. And if you remember, the vagus nerve innervates the gastrointestinal system. So acetylcholine will also have receptors on the parietal cell that helps to, again, release the hydrogen ions into the stomach. I also want to say gastrin directly has I'll go back up, has the receptors to influence the release of hydrogen via that hydrogen pump up here. Remember the hydrogen pump. We'll be coming back to that one. So also some things that will induce the release of histamine are some disease states like mast cell tumors, anything that would influence the release of acetylcholine. So organophosphate poisoning stimulates the parasympathetic nervous system. And so these are mast cells. Hopefully you've seen some of those in your ClinPath class. When animals have mast cell tumors, we will typically put them on anti-ulcer drugs because we're anticipating that they're going to have that they will have ulceration. I'll also let you know that some of our semi-domesticated species, such as llamas and alpacas, they do not do well in hospital settings. And so when they are in a place where they're not with their herd buddies, they will sometimes be very stressed. And so part of the treatment for whatever they're in for will be to treat in advance of gastric ulceration. We just go ahead and treat them as though they have ulcers. That is the same with foals. So foals being, quite often they accompany the mare into the hospital if there's some condition that she's being treated for. And if we have foals in the hospital, we'll go ahead and treat them as well for ulcers. They're little ulcers waiting to happen. So that's kind of the basic mechanism behind the stomach that we're going to talk about, how we manipulate these things with drugs.